Okay, so earlier today we talked about sometimes one of the most difficult things is to get things through legal, especially PowerPoints for a meeting that's taking place in Boston. Ajay had that experience, so he's ad-libbing.
Ad-libbing, that's right. So, my apologies. These are slides that were approved. Almost all of them were approved previously. A new lawyer looked at it and didn't approve them. So, and then they conveyed it to me this morning. So, my apologies, but hopefully I'll make it worth your time.
While AJ's setting up, I'd just like to say AJ's at BMS. He's an executive leader in digital health, scientific research informatics, research IT. He is at BMS, he's responsible for IT-related translational medicine. He's accountable for several projects, the largest being a FAIR, enabling data integration and analysis platform for research, clinical, and real-world data. I had the privilege of working with AJ back in Biovia days. I think it was called Accelerus at the time. He's also worked at Pfizer. He has not worked at NIBR. No. He's worked at the City of Hope and Elan. So welcome, AJ, and I'm sure it's going to be interesting.
I did interview at NIBR at one point, though, but I had just started. But I had just started at Elan a month before that, so I didn't go to NIBR at the end. So there's some NIBR connection here. If you're in Boston, you have to find one. So I will, as I said, I'll try to make it worth your time. At least the good thing is that a lot of the stuff came out. Coming? Okay. Well, so I do. So I started. When Margaret and Tina reached out to me, Margaret, we intersected at Accelerus at one point or the other, and they reached out to me, and I had to give some titles. So my role right now at BMS is I'm the digital lead or IT lead for cell therapy, as well as for clinical pharmacology and pharmacometrics. And so I've talked about this in past, which is making cell therapy data fair. And I was like, okay, well, but really I started thinking about it after I sent the title out. And I said, let me come up with an alternate title, an alternate way to look at the research IT or research informatics. And I came up with this alternate title, which says, when fair becomes unfair, and how are we attempting to make it fair? Because fair is right now a buzzword. Everyone has been using it for the last 7-8 years. We've been talking about fair data in some form or the other since I was working at Elan at one time in 2007-8 and before that as well. But really, we have not made that much of an headway. So when I started thinking about it, what is the problem here? There's multiple problems, and I don't know why this one is fading in and out, but it doesn't matter. So the aspiration that we have, and again, I wish you had the slides in front of you, but as I said, the aspiration we have is that we want to make data, we want to do research that is predictive. Because research costs a lot of money, and we don't want to, to take a drug out, $1.4 billion, $1.8 billion, pick your favorite number, 8 years, 10 years, or accelerated research, 6 years, unless we have a great success story like Regeneron and can bring a vaccine to market like Pfizer and Regeneron and bring the vaccine to market in months. It's really a much more prolonged process. So the aspiration is to really speed up this drug pipeline, bring drug to market faster for our patients, but there are challenges. What challenges that is, it's not the data challenge. So if anyone says it's a data challenge, I don't think it's sitting in this room, because you guys all know the real challenge is not with the data as much as it is the value that you're providing to the people who are generating the data. And that is really the theme of my presentation, that it's not a challenge with the data as much as really, if you dig deeper and deeper and deeper, you realize that the real problem is with people who are generating the data, who have no incentive to generate the data. So I'll come to that in a minute. So the approach you want to take in research informatics and research IT is a lab-centric data approach to generating the data that you need for predictive research. So you kind of flip the script, and you say, what do I need to do to even make the data fair? And what you need to do is, how do you incentivize the people who are generating the data? And so really, then once you have that, then you can start thinking about how do you create a technology framework or technology enablement to have all these things like metadata and ontologies and semantic integration, relationship, knowledge graphs, and finally FAIR, right? So an example that I'll give is the immunology, oncology, cell therapy domain that I have been working with. So really the question is, how do I make still? Otherwise, I had a few cartoons that were actually, public domain, like Calvin and Hobbes or something like that. But even that doesn't want to cooperate today. So I don't know why. Maybe this is flaky. Okay, perfect. So this is the cartoon that, maybe this is the reason why they didn't approve. But really, people want to know, how do I make the data fair? The question is, we really have to put things in perspective. We are telling people that, hey, look, do extra work. People who are in the lab, they have to generate extra metadata, do metadata entry, but where is the incentive? They are being judged by the number of experiments they do, number of new compounds they make, number of new assays they do. They're not being incentivized to enter metadata. I remember I was working at City of Hope. I was head of research informatics and research IT at one point at City of Hope, which is a comprehensive cancer center based in LA. And I had the same discussion with people who are, people who are oncologists and who are seeing patients on a day-to-day basis. And then there are people who are clinicians, who are medical doctors, but they're also doing research. And we worked with one of the melanoma specialists, and he wanted to create a disease registry for melanoma with over 2,500 data points per, it's a sparse matrix, but 2,500 data points per patient. Now you think about an oncologist who is seeing a patient who is in stage 1, stage 2, or maybe even stage 3 and 4 melanoma. And this oncologist is seeing how many patients a day? 10, 20 patients a day, probably 20 patients a day. Now, do you want that doctor to enter all the data for a particular patient? 5 to 600 are required by state of California. Do you want this medical professional, instead of seeing more patients, saving their lives directly, or you want that doctor to see 10 patients or 5 patients a day and spend the rest of the time entering the data to make a really comprehensive disease registry that can be useful for research? That is really the question that we have, and that's a question we have in the basic research lab as well. Where do you want your experimental scientists to spend their time? And how do we align the two? And the other things, really the triangle that we always talk about is that technology is the easiest part. The next difficult challenge is really the process. And the most difficult part of the technology triangle is people. And that is the problem that we are trying to solve here. So I think I do have one more that before I go, while you guys look at this, I'll talk about some of the challenges, right? So ELNs are primarily, what are the technology challenges? The technology challenges are that ELNs are primarily, paper on glass. Primary paper on glass. So you're used to do something in a paper notebook, now you're doing it on a screen. They're meant to capture minimum amount of data. They're really not meant to, capture research, long-term data, like, multiple dozen data points per experiment. It is really, you get a lot of the templates are standardized. You try to make standardized template. We heard about some of the talks where people are trying to standardize the templates for data collection. But in some ways, it runs counter to agile research. Because if you go to and talk to, especially in cell therapy, if you go and talk to the scientists, they will say, hey, we want really, to change the assays that we are doing. We want to change the template. How quickly can you deliver a new template? Well, a day, 2 days, 3 days, and they're backlogged, right? Because you have multiple groups coming to you and saying, hey, I want a new template. You can't really keep up with a lot of that. So you kind of come up with structured way of capturing minimal data set. And it's not really comprehensive. And it's really meant for, smaller groups working together, ELNs in general. They're not really meant for everyone working together. And that's why you have all these ELNs that are chemistry specific or analytical chemistry specific or biology specific. And people have tried in recent past to bring all of these together. But really bringing together has not been that successful yet, right, in terms of data capture. And the same kind of issues that you guys all know about LIMS systems. They're gear in some ways you're capturing minimal data required. The process questions are the cost that I mentioned earlier, and really the user adoption, agility of the process, and then there are people problems. So the first thing it took me a while after I started working with people at former Juno site, and I started walking in their shoes. I started to realize that there's just things that don't work, and there's things that you don't even because we tried telling them we're going to have fair data. We tried telling them that we're going to capture metadata, and so through hard work and a lot of hard knocks, I realized that the words you don't use in front of a biologist, if you want to get buy-in, at some point you may, but you don't want to use the word fair data, you don't want to use the word metadata, you don't want to use the word semantic integration, semantic knowledge, or semantic search. Definitely don't want to use words like data warehouse, data lake, data mesh, data fabric. You don't want to use the word knowledge graph. You don't want to use, I'm going to put all your data in one place, and you don't want to say that I'm going to give you a single source of truth. Even though you want to do all of that. This is not how you start doing research informatics, because it really adds no value and it turns people off. So you start by saying that, okay, what is it that, and you start by saying, I'm going to acknowledge what you're doing, and you want to really, what you want to do is you want to come up with a framework that kind of incentivizes them. And this is really the slide I wanted to show. But if you meet me offline, I'll show it to you. But it really has no proprietary value, I can assure you. It has absolutely no proprietary value to BMS or anyone for that matter. It just helps me give a talk. But really, what you want to think about is, if you were to talk to an experimental scientist, and this is an aspiration, right? It's not that we have this system in place today. It's aspirational, we have pieces of it, and I'll tell you which pieces we have and which pieces we don't have. But you want to say that, look, you're going to plan an experiment. When you're planning the experiment, what is the system that I can give you so you can explore the previous experiments that have been done? Now think of all the technology that goes behind the scene. When someone wants to explore this previously done experiments, but all the systems we talked about today, and you guys have all worked on in the past for many, many years that you have worked in the industry, that requires an assay management system, that requires getting the data out of PowerPoints, that requires building data warehouse, or data lake, or data fabric. It requires ontology, it requires semantic search, but that is not what you're incentivizing people. You want to do a new experiment, you want to do a new assay, you want to go to a new therapeutic area. I'm going to build a system for you. What system do I build for you that will let you explore the previous knowledge? Right? The second thing you say is that as you're typing the description of your experiment in your ELN, can I go and match the metadata without using the word metadata? Can I, as you're typing, can I give you some of the previous, not just the related experiments as you're designing the experiment? I can give you the previous experiments, ELN pages, if you will, from the experiments that you've done in past. Can I give you the information about the same protein or same antibody or same same molecule from external publications, from in-house sources? And can I tell you the feasibility of that experiment? That's all. Behind the scenes, you may be calculating indices, you may be calculating all kinds of things. But the way you're framing it, the framing of the question is now different. Then as you're running the instrument, what is it that even without telling the users, and we saw a great presentation that Max gave and other people gave, but how do I, can I just continuously, or the previous speaker gave, can I continuously extract the metadata? And we have used systems like, for linking ELN, in our case, BenchLink. We have used a system, and Sapio actually, we have used both in two areas that I supported. In one place we have used Sapio, other place we have used BenchLink. And what we have done is taken the metadata, linked the experiment, extracted the metadata, put iRods on top of it, extract the technical metadata, and create a seamless link between the ELN and this. But we don't need to tell the users we're using iRods. That is not the point. The point is that we are continuously extracting the technical metadata, scientific data, process metadata, and putting it in a place where it can be used for people who are downstream users. The next thing is, can I help you monitor your experiment? And this is the place where we have done some proof of concepts, but really nothing to show for today. And that is we are using, say, during the COVID times, we said, can we experiment with virtual reality glasses? And can someone be sitting in the lab, one person and other person who is twinning with that person? Because COVID, we had only half the people or a quarter of the people in the lab at a time. They're working collaboratively by monitoring each other's experiment and things like that. We have done some proof of concepts with Internet of Things. So those kinds of things. But again, from the user perspective, your experiment is being monitored. What alerts do you want? Recording the results, same thing. When recording the results, what kind of metadata are we extracting? What kind of automated analysis are we doing for you? And finally, are we capturing, when experiment is closed, what is the kind of knowledge are we extracting from that experiment for downstream use? So this is the value proposition for the experimental scientist. Again, it involves absolutely no technology talk, right? It is, what is it the value that I am giving you? Now, behind the second category of people, who are your second category of people are people who are more of, in social media terms, you would call them, influencers, right? So these are maybe 5%, 10% of the users, and you want to influence them. There you can start becoming a little bit more technical. And I'll show you this article. This is a recent article from Wall Street Journal. I don't know how many people saw this article. This entire article, it came like 2 weeks ago, maybe? And the entire article is about why it took Apple and the resources of Apple to actually create a new app that they released this week or last week, right? A classical music app that they just released. And the entire article is about what is the importance of metadata. And the reason the music app was, it took such a long time for anyone to come up with a really good classical music app is because the way the metadata is captured for classical music is very different from the way metadata is captured for popular music. And they had to put a lot of resources in order to streamline the metadata capture. And you read that and you say, and they tell you how much, how many people and how much money and all that it costs. And you say, this is something relatable. And this is what you go to, now you understand that it's not just a problem in a lab. Even in a seemingly simple thing like music, this is a really challenging issue. How do you capture metadata? How do you use the metadata to make the choices more, make the downstream use? So we started with that, and then we said, okay, now we can explain to you not just what is metadata, which is information about data, but we can also now go in terms of explaining what is scientific metadata. And there is a paper that came in Nature, if this only works, not my day, as you can see, but this is Nature, and I can give you the reference on this paper, but this is a paper in Nature, and really nicely lays out what is scientific metadata. And if you can go to this 5% or 10% of your influencers, and you can explain to them that, look, this is why we are really doing this. And this is why we really need scientific metadata. We need metadata, we need scientific metadata, and this is the reason why we need it. And once you have that, then you can explain to them in a very simple scientific terms how you're going to connect in terms of cell therapy, how are you going to connect the expression construct plasmid to the mature protein, to the expression media, to the recombinant protein, and then eventually a project and all the entities kind of come together. But this is the approach that you can potentially take in order to explain to a scientist, which is why the metadata is important to meaningfully connect all this data. And once you have that, then you can say that not only are the metadata important, scientific metadata important, to connect the experiment, to connect the entities, but you can also now connect the different projects. You can connect not just the projects, but you can connect the in-house data with the external data, enterprise data across BMS, and outside data as well. But that requires, so now you can go towards this particular kind of chart where you can say, look, what we are really trying to do is we are trying to connect this knowledge, we are trying to derive, this semantic relationship by taking really a list of data that you may have. We are going to from there to the ontology, to the glossaries, to thesaurus, to taxonomy, to ontology. And this is how we are capturing, this is the reason why we are doing this. This is the reason why we are capturing the knowledge, which is what we did. And so what we did was we did a few projects. So a lot of the related projects that we did, but I'll come to one specific project and then I'll explain the entire ecosystem in about 3 minutes that I have. So one of the projects that we did was called Biosample 360, and now currently we are doing a project called Biospecimen 360. And what we did was we created a data model for all the biological entities. And the way we created the data model, so for example, cell line, plasmid, antibodies, protein, antigens, viruses, car constructs, car parts, things like that. The approach we took was our system in this particular case was Benchling. And so what we did was we said, okay, well, let's not worry about Benchling. Let's not worry about workflows, any of that right now. If you were to just look at, say, for example, a protein in isolation, what are the kinds of metadata you would need to define a protein? And so we referred to literature, we referred to open registration systems, vendor systems, whatever. And just these are the metadata that you would need in order to define just a protein. And then we said, okay, what are the computed properties that you would need? Then we said, okay, what are the properties that you would need if your protein was to be manifested in a lab? So what are the batch properties that you would need? And then we said, what are the properties that would link this particular protein to different projects? What are the properties that you would need to link to different entities? So we categorized into 5 categories, and we did that for all of the entities. So of course, we also did very elaborate, and I think that is why this particular presentation was not allowed, which is we actually mapped out in detail all the workflows within a cell therapy lab. We removed the specific data collection points and all that, but that is the one that I think but it was already presented last year at BioIT, so and somebody may already have a photo of it, or it may be even in the repository. But I think that is why. So we had this entire flow chart in the presentation I had, which just says how we went to each lab, found out what data goes in, what data comes out, how we can put it, each of the data, how we can contextualize with the ontology, streamline it, normalize it, standardize it, and map it out. So we did that, and then based on that, we created the knowledge graphs. But we did a couple of things that are slightly different, and that is when we created, not just we created the knowledge graphs, but we were doing a lot of different projects, and I'm going to skip a few things, but a lot of different projects. So we were doing, one of the projects that we did was within Benchling itself, we can standardize all the data, because at one time or the other, people had just created everything in the text form. So what we did was we worked with one of the companies, one of the outside companies, to actually manually, extract the data and map it to the ontologies and make sure the data, all the text was converted into structured data from the notes fields and other fields, right? So we did that. I'm sure it's bothering people, so I'm going to remove this for now. So we did that, and then we worked with another company to actually run, to take that as the training set, and then we worked with other company to actually run NLP on the notes fields of Benchling and convert it into knowledge. And we take that, and now of course all the data, new data of course, we deal differently. We took that data and then we put it on a data lake, a project that we did about 4 years ago. And from data lake, the data goes to a system called Discover with a Q. There's a company called Ontoforce, and they have their own way of doing knowledge graphs and things like that. So we actually put it in Discover. So basically what ends up happening is that now you can look at the data, the cell therapy data in context of all of BMS data, and you can also look at it in context of external data sets because they have like 160 public data sources and hundreds of BMS data sources. There's real world data there. There's other BMS projects which are there and all of that. Still not as much of discovery data that we would like to have. It's much more of downstream data. So that was the first project we did, was to take the metadata within BenchLink, standardize it. The other project we did was we took all the data from within cell therapy, whether it's from study log or one of the LIMS system or BenchLink, and harmonize the data across cell therapy. Third component is this, putting the data, all of that data, not just BenchLink data, but all of the data in Sage, and then Sage to Discover. And then we did the reverse thing. If we looked at the kind of queries that are coming in, and we say, okay, if we were to answer these questions, how would we improve the data collection templates? Because you have to kind of complete the loop. You're just going one way, right? But you want to go the other way as well. So that is the last part of the project where we, so right now what we are working on is we're doing the biospecimen 360 because biosample is done, it's in production. Biospecimen part will be done by the end of next month. So we're tackling one at a time. I think that would be probably the end of it at this stage for that phase. So that basically brings me to the end of my presentation, but I figured I'll show at least this slide. No lawyer would object to it. I think if you don't have a slide on FAIR, you kind of can lose your job. So I figured at least I'll keep that slide if it ever comes up. Hopefully it will. There you go, at least one slide. Okay, so now I've done my job. I don't think anyone in informatics can fire me because you have to kind of show something about FAIR. Even though I started by saying that nobody should ever use the word FAIR in front of a biologist especially. Okay, and I'll be glad to answer any questions you may have. See, no questions because there are no slides.
Oh, Chris. Have to at least have one question.
Yeah, I have one question. Like, this is like ACS 101. If you get ACS, make sure every presenter gets a question.
Exactly.
So I have a question about when fair becomes unfair. What I've observed is as we try to fairify the collection of data, often the scientists that are creating the data feel that they're made to do a lot more work and they don't get any additional rewards out of it. It's there to help the data scientists or to help someone downstream. How do you deal with that sort of feeling that you get, maybe not from like more senior people, but more junior people in the lab that are like, I don't want to have to do more work all the time.
That's why you have to, so that's exactly, that was the theme of this, right? So how do you automate a lot of this so that the people who are collecting the data don't feel the burden? I think it was in 2015, I think there was an article in New England Journal of Medicine which talked about data parasites. I don't know how many people read this, and this was an editorial. And the editorial said that, and it was written by someone who collected the primary data. It was, again, in hospital context, who collected primary data, and then other people did research with it and got publications and thereby got grants and all that. So they said, all the people who are data scientists are data parasites. Because all they're doing is taking my data, and then they're getting the grants based on that, and I'm not getting any credit. So basic economics, if you take any economics course, you'll know that people will do what they're incentivized to do. And if you don't incentivize people, then people will not do it, regardless of how much effort you put in. There's only so much you can do by bringing a hammer down and say, you will collect this data. Because I gave you an example in a hospital. Are you going to see patients, or are you going to collect data? So I think that is why the thrust of my presentation was that if you want to make data really fair, you have to automate the systems that they make data fair and that collect the data from the primary people, data generators. You cannot expect a fair system if you expect people to enter the data. That's the bottom line.
Yeah. What's your experience in terms of the adoption of these systems? I see like a lot of effort put into creating make the data fair in terms of data cataloging or metadata collection, but sometimes it's difficult to have the users actually using those systems, so using the enterprise searches, searching the data catalogs to get the information. What's your experience in terms of adoption? Mm-hmm.
So different experience for different types of users. So I would say that when it comes to people who are, I'll give you an example of people who are in cell therapy or people who are doing basic biology. The experience with them is still rather poor for two reasons. One is that they say that I want to use this. They're using the system maybe 2 or 3 times a year because that's all their need is. And every time they go there, they're like, no, I don't remember how I did this. Can someone go? And so they just try a couple of things. It doesn't work, doesn't give them results. Oh, that system never works, right? And so that is, I think that is, even though I think that Discover from OntoForce is a fairly easy to use system. And I think that really the good selling point is that their interface is nice. It could be easier, but it's fairly good. It's, but still people say, well, I don't remember what I did 6 months ago or 3 months ago. Or they will come across one particular data set that is still not there. And they say, oh, I really wanted this data set, but it's useless. I don't have that data set. So I think those are the kinds of challenges that we'll need to work through. And that is why I go to the second stage, which is, am I able to find the right influencers who will be there, not people in informatics, not people in IT, but people who are in their lab, people who in their group, one person who is kind of just going to say, hey, if you have any problems, you have any questions, just come to me and I'll help you out. I think that model will work. I also found that people who are downstream from the very basic scientists, people who are doing more data analysis, people who are doing work in translational medicine. So people who are more translational research, translational medicine. If you go farther downstream, people are a little more open to doing exploration and using and all that. So I think there's a gradient there. And yeah.
Okay, great. Thank you.
Okay.
Thanks, Ajay.
Thank you.